Thank you very much, Ting, for that nice introduction, and thanks to the Department of Bioengineering for the invitation, and thank you all for coming out. This is a lovely audience, and I hope I can entertain you for the next 50 minutes or so with tales of eukaryotic cell cycle control in yeast. So I should give some credit to NIH, General Medical Sciences, for supporting this work pretty faithfully for the last 10 years or so. So let me start with physiology, what the cell cycle is all about, what we're trying to understand, and then I will talk a little bit about the molecular biology that underpins this physiology, and then we'll talk about the general principles of cell cycle control and mathematical modeling. So the cell cycle is the sequence of events by which a growing cell replicates all of its components and divides them more or less evenly between two daughter cells, so that the daughter cells inherit the machinery and the information that's necessary to repeat the process. As you probably know, the cell cycle is central to everything in biology because all cells come from previously existing cells by this process of cell replication and cell division. So all living organisms are made of cells, and all cells come from previously existing cells by the process of the cell cycle. So understanding the molecular control of the cell cycle is truly a central problem in the biological sciences. And it's also of great importance in the health sciences because, as you know, cancer, thank you very much. Cancer is a set of diseases which are all related to misregulation of cell growth and cell division and cell death pathways. But one may ask, if we're really interested ultimately in understanding cancer cells and how to treat cancer cells, why would we start with budding yeast cells and spend so much time understanding budding yeast? Because budding yeast cells don't get cancer, budding yeast cells don't pay big hospital bills, and things like that. However, you're all engineers, and you know if you wanted to be a car mechanic, where would you start? Would you start with an old Beetle, or would you start with a $100,000 Audi sports car, right? Of course, you'd start with the old Beetle, and you'd tinker with that and understand how it works. And the sports car is a kind of direct lineal descendant, and all the basic powertrain and control systems are there in the Beetle. That's what you want to understand. And that's why we want to understand what budding yeast cells are doing. They're a wonderful type of cell for doing experiments, for gathering great data sets. And what we learn from understanding control in budding yeast cells carries over quite directly to mammalian cells. But I'm not going to talk about mammalian cells at all today. Okay, there are two aspects of the cell cycle that I want to focus on in this lecture. The first is what we call the alternation of DNA synthesis and mitosis. Okay, so the most important molecules in the cell that need to be replicated and divided to the daughter cells are the DNA molecules, the hereditary molecules, the genome. And during the eukaryotic cell cycle, each DNA molecule is replicated during a particular phase of the cycle, the phase of DNA synthesis, or S phase of the cell cycle. So in the cartoon here, let's see, how do I get the... Yeah, okay. In the cartoon, that would be the cell membrane and the nuclear membrane and two chromosomes in the cell, a blue chromosome and a red chromosome. And during S phase, those chromosomes are replicated, and that was probably an important piece that just fell out, right? It's under your foot. I think that's what... Okay.
Okay.
Oh, that's for changing the...
Yeah, for changing.
Oh, I don't need that, do I?
Okay, just take it out.
Okay. Where was I? So During S phase of the cell cycle, the DNA is replicated. Each double-stranded DNA molecule is copied, so you get two identical double-stranded DNA molecules. They're called sister chromatids. So at the end of S phase, there are two blue DNA molecules, there are two red DNA molecules. They're identical molecules, and they're held together by glue. That's the yellow stuff in between. Okay, so the sister chromatids are glued together during S phase. Then later, after a gap, the cell goes into mitosis, and in mitosis, those sister chromatids are to be partitioned to the two daughter cells. Each daughter cell gets one and only one copy of each DNA molecule. And the way that works, you probably know, is the cell builds a mitotic spindle. So there are two spindle poles, and from the spindle poles come out threads called microtubules, and those microtubules capture the replicated chromosomes at particular locations in the center called kinetochores. The microtubules capture the chromosomes, and the idea is that one sister chromatid is to be attached to one pole of the spindle and the other attached to the other pole of the spindle. That's a proper alignment of the replicated chromosome on the metaphase plate. When all of the chromosomes are properly aligned on the metaphase plate, then a signal is set out to continue through the cell cycle, and the next thing that happens is that the glue that's holding the chromosomes together, the glue is dissolved, and then the microtubules can separate the sister chromatids, and they're carried to opposite sides of the cell, and new nuclear membranes are built around those chromosomes, and the cell divides down the center, and you get two daughter cells that are ready to repeat the process. Okay? So it's very important that this process of copying the DNA and then partitioning the sister chromatids to the two daughter cells, that those processes alternate as you go through the cell cycle. Okay? It's just like you have a Xerox machine and a collator. Okay? So you put your paper into the Xerox machine, you get two copies of every page, then you put it into the collator, and the collator separates it. So you get one complete book. Okay? And those occur at different phases in the cell cycle. So this is the Xerox machine, and this is the collator. And you have to go around the cycle, first copying and then collating, then copying, then collating. It's very important that you don't, for instance, slip back. So you go from the G1 phase where the chromosomes are unreplicated through S phase, the chromosomes are replicated. You don't want to slip back and do DNA synthesis again, because then you'll get 4 copies of every chromosome. And that's not a good idea. It's not a fatal mistake, but cells don't like to be polyploid like that. Even worse would be if you go into mitosis and separate the chromosomes and come into G1, and then you slip back and you try and go into mitosis again without doing DNA synthesis. That's a fatal mistake, and the cells will die if they do that. You can mutate cells so that they make these mistakes and they're very unhappy or they're dead, but it's very important for the normal control system to assure that there's irreversible progression around the cell cycle, G1S, G2M, G1S, G2M. So that's one thing I want to understand, why this irreversible progression. Yes, question?
So things like Trisomy 21 and things like that, that happens in the metaphase to telophase, right? They don't come apart and then
yeah, so trisomy 21 actually happens during meiosis, not during mitosis. Meiosis is a different process where the homologous chromosomes have to be separated as you form the germ cells. It's a very similar, but it's also different, okay? So if there's a mistake there, then you get the trisomies. Yeah. Okay. The other question I want to address is, you need a molecular control system that's going to organize these events in time and make sure that you get this alternation and you get these irreversible transitions. But that molecular control system has to function reliably in the face of unavoidable molecular noise. Because let's face it, a yeast cell is very small. And it doesn't have very many copies of the molecules that have to implement this control system. Okay? So it's It's not like a chemical reactor with a mole's worth of substances. You only have one copy of every gene. You have a handful of copies of messenger RNAs. You might have 100 or 1,000 copies of the proteins. So there's bound to be stochastic fluctuations in this control system, and we want to make sure that it operates reliably in the face of that kind of noise. So that'll be the second half of the talk. Any other questions to start? Okay, so that's the basic physiology. That's what we would like to understand. Now we have to learn a little bit about the molecular biology. What are the underlying proteins that control the events of the cell cycle? In all eukaryotic cells that have been studied in any detail, it's always the same group of proteins. These proteins are called cyclin-dependent kinases, CDKs, cyclin-dependent kinases. These are protein kinases. That means they are enzymes that phosphorylate other proteins. Okay, so if I'm a protein kinase and you're a target protein, I add a phosphate group to you. And that phosphate group carries a lot of negative charge. It causes the phosphorylated proteins to change their shape and to change their activity. So when the target protein gets phosphorylated, it can be turned on or it can be turned off. Okay, so the way the cyclin-dependent kinases control events in the cell cycle is by phosphorylating proteins involved in DNA synthesis and later phosphorylating proteins involved in mitosis. And whatever is done in one phase of the cell cycle has to be undone later because it's a repetitive process. So wherever there's a kinase putting phosphate groups on, there's a phosphatase enzyme later taking the phosphate groups off. Okay? The cyclin-dependent kinases, as their name suggests, require a partner in order to be active. Those partners are called cyclins, and in yeast, the cyclins are called CLIN2, CLIB5, and CLIB2. Okay, those are the cyclin partners of the kinase. These partners drive the very earliest event of the cell cycle, which is called start. It's when the yeast cell commits itself to a new round of DNA synthesis and division. CLIB5 dependent kinase is important in driving the events of DNA synthesis, and later CLIB2 dependent kinase drives entry into mitosis and blocks exit from mitosis, blocks the metaphase to anaphase transition when the glue is dissolved. Okay? So it's the rise in activity of these cyclin-dependent kinases that drives the cell out of G1 into S phase and then into M phase and causes the replicated chromosomes to align on the metaphase plate. In order to finish the cycle, we have to get rid of the cyclin-dependent kinases and dephosphorylate all the proteins. And there we need a different set of components. So there's a protein complex called the anaphase promoting complex. The anaphase promoting complex essentially promotes the dissolution of the glue, which allows the sister chromatids to be pulled apart. That's called anaphase. So it promotes anaphase by dissolving the glue, and it promotes exit from mitosis by degrading the B-type cyclins, CLIB2 and CLIB5. Okay, so it gets rid of the things that brought you into mitosis to allow you to get out of mitosis. The anaphase promoting complex needs a partner to direct its activity to the right places. That partner is called CDC20, which appears very abruptly as cells exit from mitosis. And then later, CDC20 is replaced by a different partner called CDH1. But basically, they do the same thing. And finally, in G1, there's a stoichiometric inhibitor of the cyclin-dependent kinases, which can bind the cyclin-dependent kinases and keep them inactive. I'll say a little bit more about these in just a second. But are there any questions so far? Okay, a lot of new friends that you're just meeting. I hope you get the basic idea. Cyclin-dependent kinases drive you into S phase up to metaphase, and then you have to get out so you can start over again. Okay. What did I do? Sorry. Now, we know a lot about the molecular biology of the interactions amongst these proteins, these regulatory proteins. And this is how we represent that interaction network. If you were an electrical engineer, it would be a wiring diagram for a VCR or something like that. We're bioengineers. Our components are these different protein complexes. The solid arrows are chemical reactions. So this describes the synthesis of CLIB2. It combines with the kinase to form an active protein kinase. And this is the degradation of CLIB2 here. And the degradation of CLIB2 is driven by these two components, CDC20 and CDH1. And then there are phosphorylation and dephosphorylation reactions. So for instance, the CLIB2 dependent kinase can phosphorylate CDH1 and inactivate it. And there's a phosphatase called CDC14 that can dephosphorylate CDH1 and activate it. Okay? So that's how you read a diagram like this. These are each individual biochemical components. They have reactions that produce them and destroy them, and they're enzymes, so they catalyze other reactions in the network. It's a very dense network with lots of feedback and feed forward, positive feedback loops and negative feedback loops. And our job is to try to determine whether this mechanism can account for the known features of cell cycle regulation in budding yeast. Okay? I don't think it's obvious from looking at the network. And as a matter of fact, the network, I think, is so complicated that I would be hesitant to rely completely on my intuition to figure out how a network like this is going to behave. Okay? And what we've been trying to do for many years now is to convert mechanisms like this into mathematical models, be very precise about our assumptions of how it works, and then do analysis and computation on those models, compare to experiments, and see whether this network really is consistent with the way yeast cells behave. Think of this as a hypothesis, right? We don't know that this is true. We think it's true. How do you tell whether it's true? Well, it needs to fit the facts. Harry, did you have a question?
Well, I said, because you already indicated with a low number of these molecules in the cells, so there's sort of a potential for all of these reactions to occur, but there's a physical coincidence in space that has to be present.
Right. So, yeah. Right, so we'll get to that, okay? But first I want to do a deterministic model with differential equations, and then we'll convert it into a stochastic model. Alex?
And is the translation of genes into these proteins and the regulation of the genes part of that network?
Yes, it is. So, for instance, MCM1 is a transcription factor, and that little dashed arrow means that's the transcription factor that activates the gene that encodes CLIB2. Okay, so there are a number of transcription factors here that aren't, and there's messenger RNAs that I haven't even indicated on this diagram. Okay. So I want to start with deterministic models and give credit to my collaborators, my longtime collaborators, Bela Novak and Kathy Chen, who've been working on this project from the very beginning, and two postdocs. They're now established scientists in their own right, Attila Cekasnaj and Andrea Ciliberto, who were involved in this from the beginning. Okay. So the way I want to describe how this network functions is to divide it into kind of simpler modules and try to convince you that there are little regulatory modules here that basically control the properties of this regulatory network. And I want to focus in on CLIB2 dependent kinase and CDH1. Remember, CDH1 is involved in a proteolytic pathway that destroys CLIB2 and therefore destroys the activity of the kinase, because the kinase has to have a cyclin partner in order to be active. Okay? On the other hand, CLIB2 is a protein kinase that can phosphorylate CDH1 and inactivate CDH1. Okay, so CDH1 is an inhibitor of CLIB2-dependent kinase, and CLIB2-dependent kinase is an inhibitor of CDH1. So these two protein complexes are involved in mutually inhibitory connections. And you might guess that at least it's possible here to get two different states of the control system. There could be a state in which CDH1 is active and CLIB2 is rapidly degraded, so you can never get the kinase activity. And that we would call the G1 phase of the cell cycle. When CDH1 is active and there's no kinase activity, you're in the G1 phase of the cell cycle. But the other possibility is we can have lots of CLIB2-dependent kinase activity. CDH1 is phosphorylated and inactive, so CLIB2 isn't destroyed, and that would be kind of the SG2M phase of the cell cycle. So there's this basic mutual inhibition that can create a bistable switch, and those two stable steady states are the two basic phases of the cell cycle. So let's see how that would work out mathematically. Here's a simple representation of the motif. You have synthesis and degradation of CLIB2, you have phosphorylation and dephosphorylation of CDH1, and CDH1 is degrading CLIB2. Now that looks a lot simpler, and we can just translate that directly into a pair of nonlinear differential equations for the amount of CLIB2 dx dt and for the activity of CDH1. Okay, and on the right-hand side, we have the synthesis of CLIB2, that's this arrow, and we have the degradation of CLIB2, that's this term, and the degradation depends upon the activity of CDH1. Similarly, the activity of CDH1 depends upon the kinase, which is phosphorylating it, so that's the negative term because it's inactivating CDH1. And there's a phosphatase, represented by A, which is catalyzing the reverse reaction, taking the phosphate group off and activating CDH1. Now, there are many ways to analyze a pair of nonlinear ODEs like this. I'm just going to skip right to what a mathematician would call a one-parameter bifurcation diagram and what a cell biologist would call a signal response curve. Okay? The signal in this case is the ratio of the activity of the transcription factor S that's driving the synthesis of CLIB2 to the activity of the phosphatase, which is trying to activate CDH1. If I treat that as a parameter for these differential equations and plot the steady state activity of the CLIB2-dependent kinase, that's species X, because of this double negative feedback loop, I get a bistable system. There's an off state to the control system, and there's an on state to the control system. There are two saddle node bifurcations, and there's a region of bistability in between. So for certain values of the parameter, this switch can be either off or on. Okay? So that's a basic property of this little network. Now, how do we induce the switches? We can induce a switch from the G1 phase of the cell cycle into the SG2M phase of the cell cycle by increasing this parameter, going past the saddle node bifurcation point, and then we'll automatically turn the switch on. Okay, so that's starting the cell cycle. And then to exit the cell cycle and go back to G1, we have to increase the activity of the phosphatase, drive the parameter in the other direction, go past the other saddle node bifurcation, and we can flip the switch off. Okay? So it's a bistable switch, and we want to alternatively flip the switch off and flip the switch on. Okay? Just like the light switch on the wall is a bistable switch, and you come in, you have to push on the lever, and you flip the lights on, and when the seminar is over and you leave, you push it in the other direction and you flip the switch off. Two stable, steady states. So if you're with me this far, then the next question is, what are the mechanisms that flip the switch on and flip the switch off? Yeah, Harry.
It is true that there's a bistable situation with a light switch, but I don't have to go all the way down to where one equilibrium ceases to exist, which is what you show here. If I push hard enough, I can leave in the middle of that bistable region. So why does that not conceivably happen?
So I think what Harry is saying is there are two ways to flip a switch. You can flip a switch by pushing the parameter past the saddle node, and then it'll flip automatically. You could get this switch to flip if you could perturb it past the unstable steady state, and then it would flip. Okay? Actually, that's not what you want to happen in a cell. because that would be a kind of spontaneous flipping of the switch due to molecular noise. And as a matter of fact, what I want to prove to you later is that won't happen. Given the level of molecular noise in a yeast cell, the switch won't flip by accident. You have to push the bifurcation parameter. Okay, so back to the control network. We have the double negative feedback here that creates the bistable switch. Now I need some interactions that'll flip the switch on. The switch is flipped on by Cyclin-2 dependent kinase, because Cyclin-2 dependent kinase can also phosphorylate and inactivate CDH1. So the Cyclin-2 dependent kinase, the starter kinase, is an inhibitor of CDH1. And when it pushes down CDH1, then CLIB2 dependent kinase can come up. And CLIB2 dependent kinase comes down here and inactivates the transcription factor that's driving the production of CLIN2. And CLIN2 is a very unstable protein. So when CLIB2 turns off its production, CLIN2 goes away. So CLIN2 is a friend of CLIB2, but CLIB2 is an enemy of CLIN2. And the double negative feedback loop is embedded in a 3-component negative feedback loop. Minus, minus, minus. Now, what are the consequences of that topology? Here's the motif. What are the consequences of the topology? I want to consider CLIN2 as essentially a bifurcation parameter on the underlying bistable switch. When CLIN2 is 0, I can have 2 stable steady states with CLIB2 dependent kinase low, with CLIB2 dependent kinase high. But we start in G1 phase of the cell cycle. We start in that steady state. In order to induce the switching of the control system, we have to create some CLIN2. And as CLIN2 is produced, it drives the system past the saddle node bifurcation, flips to the on state. Now CLIB2 kinase activity is high. It shuts off the production of CLIN2. CLIN2 goes away, but now we're captured in the upper steady state. And now you see that the transition from G1 into S is an irreversible transition. Okay? You need a signal to flip the switch, but once you flip the switch, the signal can go away and the switch holds in the upper steady state. That's why the transition is irreversible. Okay? You with me? So, that commits the cell to doing DNA synthesis and doing the beginning of mitosis all the way up to metaphase when the chromosomes are aligned on the metaphase plate. Now how are we going to get out of mitosis and back to G1? That's the job of a different part of the control mechanism. In fact, it's the job of this phosphatase CDC14, because CDC14 takes the phosphate group off and activates CDH1. So CDC14 is a friend of CDH1. Now, how is CDC14 linked to CLIB2? I'll let you sort it out, but the end result is that high activity of CLIB2 eventually activates the phosphatase CDC14, which activates the very thing that kills CLIB2. So again, we have a bistable switch embedded in a negative feedback loop. But this negative feedback loop is responsible for the other major transition of the cell cycle. Now, again, a bifurcation diagram, but a different bifurcation parameter. Now the activity of the phosphatase. And in order to get out of G2M, we have to do a transient release of the phosphatase CDC14 so that the cell can get driven past the saddle node bifurcation, flip to the lower state, and CDC14 has done its job. It can go back into its hole and not bother me anymore, because now I'm back in the G1 state of the cell cycle. You with me? That's how it works. Questions?
What
Ting.
if the, sorry, I have a question.
Yeah.
What if the small double negative feedback is not in the big loop? How the dynamic will change?
Well, if it's not embedded in the big loop, then the big loop can't affect it, right? The function of the negative feedback loops is precisely to flip the switch and then to go away. Once the switch is flipped, because it's a negative feedback loop, the signal goes away.
Okay.
Okay? And that's important. Okay, so the bistability creates the possibility of irreversibility, creates the two phases, the possibility of irreversible switching. And the negative feedback loops are just there to flip the switch one way or the other.
I see.
So, let's put the two pieces together. I put the bistable switch in the center and the two negative feedback loops on one side or the other, and put the two bifurcation diagrams back to back. So here we start in G1. Well, let me say the kind of neutral condition is no Clin2, no starter kinase, and no phosphatase, CDC14. The neutral position is here in the center. And in the neutral position, we see the bistability. You can be in either one or the other state. And we start in G1, so there's very low CLIB2 dependent kinase activity. And now the cell cycle is just moving around these hysteresis loops. So we go from G1 into S and G2 because you turn on the starter kinase. Then the starter kinase goes away, you go into mitosis, and in order to get out of mitosis and to go into anaphase and telophase and back to G1, you need to activate the phosphatase. Okay? So that's our picture, our theoretical picture of the cell cycle, with the various phases of the cell cycle mapped onto the bifurcation diagrams. And now you understand, I hope, why you always get an alternation. You always do S phase, and then you do M phase, and then you do S phase, and then you do M phase. It's an irreversible progression built into the topology of the control network. Now, if this is true, then we should be able to get some direct experimental evidence for the bistability of the control system in the neutral state. If we were to knock out both Clin2 and CDC14, in principle, the remaining control system can be either on or off. And we would like to design an experiment by which we can control which state it's going to be in. Okay? And these experiments were done by some of our yeast colleagues, yeast molecular geneticists. In the one case, the geneticists used a different cyclin called Clin3, which they could control. Okay? So now this is an experimental system where the experimentalist is controlling Clin3. And the prediction is if you just expose the cell to a small perturbation with Clin3, pushing it in this direction, and then remove Clin3, it won't be enough to flip the switch. So you'll see Clin3 activity go up, and then you'll come right back to the original steady state because you haven't gone far enough to flip the switch. But if you perturb the cells a little bit more with Clin3 to get past the saddle node bifurcation, then you'll see the switch flip. Okay? And that's the experiment that was done by Fred Cross at Rockefeller University some time ago, 10 years ago. Published in Molecular Biology of the Cell. He did exactly that experiment and proved that there are two stable steady states of the system, exactly as predicted by the bifurcation diagram. And in fact, this experiment was done to test the prediction of the model. In fact, the title of the paper is Testing a Mathematical Model of the Cell Cycle. And in fact, it's the first time that a molecular geneticist actually paid attention to what theoreticians were saying and did the experiment that needed to be done. So we have great respect and admiration for Fred Cross, a good friend. Similarly, you can test bistability on the other side by transiently expressing CDH1 activity. Remember, CDH1 destroys CLIB2. It's not a phosphatase. It destroys CLIB2. So if you express CDH1 transiently, you destroy some CLIB2. If you don't destroy enough of it, you'll go out of mitosis and then come back into mitosis. So it'll be a reversible process. And if you destroy a little bit more, then you go a little bit further, you go past the separatrix, and then you'll make the flip into G1, and you'll stay in G1, okay? And that's the experiment that was done by Frank Ullman at the Cancer Research UK in London, and published in 2009, and got exactly these results. Okay, you turn on a little bit of C80H1, it looks like you've gone out of mitosis, and you turn it off, and the cells go right back into mitosis. It's a reversible process. And you do it a little bit longer, a little bit more CDH1, and then they can make the irreversible flip into G1. Okay, I'm not going to describe the experiments to you. They're quite complicated. Questions?
The mathematical model here again has a saddle node, or you're answering a separatrix. Is this slightly different?
It's the same model, but I should say that the logic here is just a hand-waving logic. If it really is a bistable switch, it should have this property. We can't actually calculate exactly how much you're going to have to do.
Right,
Okay.
but I meant if you were to write down the differential equations for this
yeah.
system, then it is one with a stable branch and unstable
Oh
branch.
yeah,
And those
oh yeah. These are all hand-drawn pictures, but yeah, underneath are real bifurcation calculations. Okay, you with me so far? So I've presented a kind of picture of a yeast cell as a little control system, just like an engineer would be used to, with positive and negative feedback loops, with bistability, with bifurcations. And I tried to show you that the bifurcation structure of this control system is completely consistent with the absolutely crucial physiology of the control of the cell cycle in yeast cells. Okay, and it's the bistability of this control system that accounts for the irreversibility of the basic transitions in the cell cycle, and ensures, if it's working properly, that you'll always alternate S phase and M phase, S phase and M phase. Now the next question I want to address is, will this really work in the small confines of a yeast cell, which has a volume of about 40 femtoliters, which is 40 times 10 to the minus 15 liters. That's a pretty small drop. Okay? And it's not big enough to hold very many molecules, and therefore, if these molecules are trying to implement this control system, there's going to be unavoidable molecular noise, which can perturb the system. And the basic question is, will this bistable switch hold in the face of these fluctuations, or will it flip prematurely by the mechanism that Harry proposed a few minutes ago, okay, due to these random fluctuations? In order to address that question, we have to convert the deterministic model based on ODEs into a realistic stochastic model based on individual molecules and propensities for reactions to occur. And we have to do reliable stochastic simulations on that model. So that's what I want to do next, but before I do it, let me say a little